Welcome to BioBits and Bytes. Join us for the deep dive, where we take complex topics, pull apart the sources, and figure out what you really need to know.
And today, we're plunging into something that uh, genuinely feels like it belongs in a futuristic movie. We're talking about the incredible potential of tiny magnetic particles in medicine.
It really does, doesn't it? I mean, think about it. Using magnetism, this invisible force, right? And coupling it with materials measured in billionths of a meter
Uh-huh.
to navigate the incredibly intricate landscape of the human body for healing. It's pretty mind-bending stuff when you stop and think.
Absolutely. Imagine being able to guide medicine, like precisely to where your body needs it most, maybe a tumor hidden deep inside somewhere.
Or even helping damaged nerves regrow, reconnect.
Exactly. Using nothing but these tiny magnets and, well, carefully applied magnetic fields. It sounds like science fiction. But as you said, it's rapidly becoming reality thanks to these magnetic nanoparticles.
And the science behind it, the papers we've looked at for this deep dive, they really lay out a powerful vision, don't they?
They certainly do.
Yeah.
We've got comprehensive reviews covering the fundamentals, what these particles are, their unique properties, how they're engineered. But we've also got specific research digging into some really exciting cutting-edge advances.
Like what specifically?
Well, particularly focusing on how magnetism and these nanoparticles are tackling two really significant challenges. First, promoting peripheral nerve regeneration, helping nerves outside the brain and spinal cord heal.
Okay, that's huge.
And second, maybe one of the most persistent hurdles in all of medicine, getting treatments across the blood-brain barrier, to treat conditions of the central nervous system.
Right, the brain's fortress. So our mission today is basically to unpack what makes these nanoparticles tick, right? Understand the properties that make them so special for medicine.
Uh-huh. And how scientists actually make them and, prepare them so they can work safely and effectively inside us. It's not trivial.
Definitely not. Then we'll explore these applications, getting drugs into the brain, helping nerves heal. And just as importantly, we need to talk about the challenges. What are researchers still wrestling with?
Exactly. The hurdles to getting these therapies from the lab into actual clinical use. It's a journey, like you said, right at the intersection of, well, physics, chemistry, nanotechnology, and biology.
So let's jump right in. First things first. What exactly are magnetic nanoparticles? Let's break that down.
Okay, good starting point. So nanoparticle just tells you about size, right? We're talking incredibly small particles measured in nanometer.
And a nanometer is, remind us.
A billionth of a meter. So to put that in perspective, a human hair is maybe 80,000 nanometers wide. We're looking at particles typically ranging from maybe a micrometer, which is 1,000 nanometers, down to just a few nanometers across. Way, way smaller than a cell.
Wow, okay, profoundly small then. So what makes them magnetic, and why is that special at this scale?
Right. So a magnetic nanoparticle, or MNP, is special because it actually holds on to magnetic properties,
Right.
even at this tiny, tiny size. Not all materials do that when you shrink them down.
Why not?
Well, magnetism fundamentally comes from electron spins, these subatomic properties.
Hmm.
In most materials, these spins are all randomly oriented and cancel each other out. But in magnetic materials, they can align. And MNPs manage to keep enough of that alignment going, even when they're incredibly small.
And the sources really emphasize one particular magnetic property for biomedical use, superparamagnetism. That sounds important.
It is absolutely critical. This is where the nanoscale size really changes the game. Your fridge magnet, it sticks there because it's a ferromagnet. It stays magnetized even after you take away the magnetizing field.
Right, permanent magnet.
Exactly. Superparamagnetic nanoparticles are different. Below a certain size, and for the iron oxides we'll talk about, that's typically less than 50 nanometers or so.
Yeah.
They only become strongly magnetic when you put them in an external magnetic field.
Uh,
The moment you switch that external field off, poof, they lose pretty much all their overall magnetism almost instantly.
so they're like temporary magnets, on-demand magnets?
That's a great way to put it. On-demand magnets. And that's invaluable for using them in the body. Why? Because if they were permanently magnetic, like tiny fridge magnets, they'd just clump together the moment you injected them. They'd aggregate.
Which would be bad, blocking blood vessels, causing problems.
Exactly, a disaster. Superparamagnetism means they stay nicely dispersed in your blood or tissues when there's no guiding magnetic field applied. They don't stick to each other. That stability is crucial.
Okay, so that explains terms like superespions, superparamagnetic iron oxide nanoparticles, and USPions, ultra-small spions. It's about that behavior and that size, generally under 50 millimeter for the ultra-small ones.
Precisely. And iron oxide nanoparticles, or IONNs, are really the workhorses here for biomedical stuff. We're talking about specific forms of Well, essentially rust, like magnetite FeOO and magmite FeOs.
Why iron oxides? Why not other magnetic metals?
Good question. Part of it is biocompatibility. Our bodies already have pathways to handle iron, right? We need it. So generally, iron oxides are considered less toxic than nanoparticles made from, say, cobalt or nickel, although those are being looked at too. But for in-body use, iron oxides are currently the main focus because of that better safety profile.
Okay, even if fancy rust still sounds a bit weird to put in your body.
It does. But remember, these aren't just flakes of rust. They're highly engineered, often coated particles. The focus on iron oxides is definitely a key theme in the sources we looked at.
Got it. So we have these tiny, usually iron-based particles exhibiting this crucial super paramagnetic behavior. Now, since we want to use them inside us, in that complex biological soup, they can't just be bare magnetic cores, right? How do scientists actually make them and, maybe more importantly, prepare them?
Right, this engineering step is absolutely critical. How well they work, how safe they are, it all hinges on how they're designed and prepared. You need incredible control. The goal, as the sources put it, is to get shape-controlled, relatively stable, and narrow-sized distribution MNPs.
Meaning you want them all to be pretty much the same size and shape and stable.
Exactly. Predictable behavior. You don't want a random mix.
So how do they achieve that level of control? What are the manufacturing techniques?
Oh, there's a whole toolbox. The sources mention things like coprecipitation, microemulsion methods using sound waves, sonochemical synthesis microwaves, thermal decomposition, chemical vapor deposition, hydrothermal methods, even using biological route with bacteria.
Okay, lots of options. Which ones are common?
Coprecipitation is maybe one of the most common and relatively straightforward. Basically, you mix iron salts, usually iron 2 and iron 3 in a solution. Then you add a base, like ammonia. Boom, nanoparticles precipitate out.
Sounds simple, but you said control is key.
Ah, yes. The simple part is deceptive. The exact conditions are everything. The temperature, how long you let it react, the specific iron salts you start with, the precise ratio of iron 2 to iron 3, the pH, the saltiness of the solution. Changing any of those changes of the final particles, you get their size, their shape, how well they stay dispersed.
So it's like nanoscale chemical cooking, following a very precise recipe.
That's a perfect analogy. It really is. Yeah. Tiny tweaks make a big difference. For instance, one source mentioned using ultrasound during coprecipitation helps make magnetite particles that are more uniform and disperse better. That's obviously good for biological use.
Okay, so they make the core particle. But then the sources talk a lot about coatings and surface modification. Why wrap these tiny magnets in something else?
This is arguably just as important as making the core correctly, especially for biological applications. A bare magnetic core, like iron oxide, it tends to want to clump together, especially in salty fluids like blood or with all the proteins floating around.
Right, aggregation again.
Exactly. Plus, a bare surface can interact randomly with cells and proteins, which might trigger an immune response or just get the particle cleared out of the body too quickly.
So the coating is like a shield or a stealth cloak.
Kind of both. It does several key things. First, yeah, it dramatically improves stability and dispersibility, keeps them from clumping. Second, it can reduce those nonspecific interactions, maybe helping them circulate longer or avoid being instantly eaten by immune cells. And third, and this is really important, the coating provides a surface that scientists can then chemically modify.
Modify how?
Think of it as a core shell structure. You have the magnetic core, then this coating layer wrapped around it. The shell can be made of different things.
Like what materials do they use for coatings?
All sorts. Inorganic stuff like silica, basically glass, or even gold. But polymers are really common. Things like PEG, polyethylene glycol, that's a big one, used in lots of medicines already. Also polyethyleneamine, polylysine, polyacrylic acid, various combinations called copolymers, even complex branch-like structures called dendrins.
And how do they get the coating onto the particle?
Different chemical techniques. Silanization puts on silica-like layers. You can graft polymer chains onto the surface. Sometimes they talk about polymer brushes where the polymers stick out like bristles, or even simpler things like adding dispersants like sodium citrate or oleic acid right during the synthesis, which helps keep the particles separate from the start by coating them loosely.
It sounds like the surface chemistry is really where the customization happens.
Absolutely. And things like the surface charge, whether it's positive or negative, also play a role in keeping particles dispersed through electrostatic repulsion.
The sources also mention functionalization using functional moieties. Break that down for us.
Right, so that builds directly on the coating. The coating layer isn't just a passive shell. It often has specific chemical groups sticking out from its surface. Those are the functional moieties. Think of them as little chemical handles or docking points
Okay, handles for what?
for attaching other molecules. This is how you turn the nanoparticle from just a magnetic bit into a functional tool.
Hmm.
You use these chemical handles to securely attach, say, the drug molecule you want to deliver.
Ah,
Or you could attach a targeting molecule, like an antibody or a small peptide, that specifically recognizes and binds to certain cells, like cancer cells, or maybe an imaging agent, so you can track where the particles go. This functionalization is what gives the MNP its specific job.
fascinating. So we've got these incredibly small, on-demand magnets, carefully synthesized, coded for stability, and then functionalized to carry specific cargo or find specific targets. It's a lot of effort. Why go through all this complexity? What are the big medical payoffs they're aiming for?
Right, the why. This brings us to the really exciting biomedical applications. The sources often frame MMPs as a kind of prototypical tool for something called theranostics.
Theranostics. Sounds like therapy plus diagnostics.
Exactly. It's a field focused on combining diagnostic capabilities, like imaging, with therapeutic treatment all in one package. MNPs are almost perfectly suited for this. Their magnetic properties make them great contrast agents for MRI scans, so you can use them to see things inside the body.
Okay, so the diagnostic part.
Right. But at the same time, they can be carrying a drug payload, or they can be used directly for therapy through something called magnetic hyperthermia, which we'll get into. So one particle potentially doing both jobs, imaging and treating.
Wow. Okay, that's powerful. What other applications are mentioned?
Well, besides the ononostics, there's magnetic labeling and separation, using them in the lab to pull specific cells or molecules out of a mixture. And then the major focus for us today, using them as magnetic vectors, basically delivery trucks.
For delivering drugs or genes, right? Let's get into that. Magnetic drug delivery.
This is where it gets really interesting. The basic idea isn't actually brand new. One source mentioned it was proposed maybe 40 years ago. The concept is elegant, really.
What is it?
You take your carefully engineered functionalized MMPs, you load them up with your drug or maybe genetic material. You inject them, often into the bloodstream. Then you use an external magnet placed over the specific part of the body you want to treat, say, a tumor.
Okay.
And that external magnetic field pulls the drug-loaded nanoparticles towards it, concentrating them right at the site of action, the tumor, the damaged tissue, wherever you need the drug to go.
So you're literally using an external magnet to steer the medicine inside the body to the right spot.
That's the core idea, yes. The magnetic field exerts a force on the particles, guiding them. The hope is to get a high concentration of the drug exactly where it's needed, hold it there while it gets released, and, crucially, minimize the drug going to healthy parts of the body. That could mean fewer side effects.
That makes sense. How is the drug actually attached to the nanoparticle?
Good question. Ties back to functionalization. There are a couple of main ways. One is covalent bonding, forming a strong chemical link between the drug molecule and the particle surface or coating.
What's the advantage of that?
Well, the sources suggest covalently linking the drug can actually improve how it behaves in the body, its pharmacodynamics. It might help overcome drug resistance mechanisms that cancer cells sometimes develop, and because the drug is securely attached until it reaches the target, it can really cut down on those systemic side effects.
But if it's strongly attached, how does it get released to do its job?
Ah, that's the clever part. The covalent link needs to be designed to be cleavable, meaning it breaks, but ideally only when it gets to the target environment. Maybe the target tissue is slightly more acidic or warmer or has specific enzymes that aren't found elsewhere. The particle arrives, senses the local signal, and then the link breaks, releasing the drug right there.
Smart, like a little timed release mechanism triggered by the destination.
Exactly. The other way is using non-covalent interactions, things like hydrophobic forces or just electrostatic attraction between the drug and the particle. These are usually simpler to load onto the particle, but the drug release might be less precisely controlled compared to those cleavable covalent bonds.
This whole targeted approach sounds incredibly powerful, especially for diseases where getting the drug to the right place without poisoning the rest of the body is the main problem.
It absolutely is, which leads us perfectly into one of the biggest problems this technology is trying to solve.
Right. If magnetic drug delivery has been around as an idea for decades and sounds so promising, why isn't it, everywhere? Particularly for treating the brain.
You've nailed it. That's the million-dollar question, or maybe billion-dollar question. This central challenge, especially when you want to treat things inside the central nervous system. Alzheimer's, Parkinson's, brain tumors, stroke recovery is the brain's own defense system.
You mean the blood-brain barrier?
The BBB. The very same. The BBB is without a doubt the single biggest roadblock for getting therapies from the bloodstream into the brain tissue itself. It's incredibly effective at keeping things out.
How does it work? What makes it such a tough barrier?
The sources give us a pretty detailed picture. At its core are the endothelial cells, the cells that form the lining of the tiniest blood vessels, the capillaries, inside the brain. What's unique about these endothelial cells is how they're joined together.
Tighter than elsewhere.
Much, much tighter. They're connected by extremely robust structures called tight junctions. Imagine the cells locking arms, forming a virtually seamless wall around the capillary. There are almost no gaps for things to squeeze through between the cells.
Okay, so that blocks paracellular transport movement between cells.
Precisely. On top of that, these brain capillary cells have far fewer of those little pores, fenestrations, that you find in capillaries elsewhere. And they also have much lower rates of transcellular transport movement through the cell itself. They just don't do much bulk transport using vesicles like cells in other tissues might.
So sealed seams and solid walls? Anything else?
Oh yes. They're also armed with efflux pumps, proteins embedded in the cell membrane, like the famous P-glycoprotein. These act like molecular bouncers. If a molecule does manage to sneak into the endothelial cell, these pumps actively grab it and pump it right back out into the blood.
Wow. Okay, so it's locked down tight, very few entry points, and it actively kicks out intruders. That's quite a defense system.
It's incredibly effective. And it's not just the endothelial cells alone. The whole setup, called the neurovascular unit, includes a supportive basement membrane layer, cells called pericytes wrapped around the capillary, and extensions from astrocytes, a type of brain support cell called glial cells. All these components work together to maintain this highly selective barrier.
So the bottom line for drug delivery is?
The sources are stark. Up to 98% of potential small molecule drugs and virtually 100% of large molecule therapies, things like proteins, antibodies, genes, simply cannot cross the BBB from the blood in meaningful amounts. That's why developing drugs for brain disorders is notoriously difficult.
You mentioned the blood-CSF barrier too. Is that part of the BBB?
No, it's a separate barrier, mainly located in the choroid plexus, which is where cerebrospinal fluid, CSF, is made. The structure's different, the capillaries there are leaky, but the barrier is formed by tight junctions between the epithelial cells surrounding the plexus. So it protects the CSF environment, but it's distinct from the BBB protecting the brain tissue itself.
Got it. Two different barriers protecting the CNS. Given how tight the BBB is, how do essential things like nutrients actually get into the brain naturally?
Good point. The brain obviously needs supplies. The sources outline the few, very specific ways things can cross. Tiny, fat-soluble molecules like oxygen and carbon dioxide, they can just diffuse passively right through the cell membranes. Simple enough.
Okay. What about bigger things like glucose?
Essential nutrients like glucose, amino acids, usually things smaller than about 500 Daltons. They use specific protein channels or transporters embedded in the endothelial cell membranes. This is called carrier-mediated transport, or CMT, like dedicated doorways for specific molecules.
And for even larger essential molecules, like hormones or specific proteins.
For those, typically larger than 600 Daltons, the brain uses a process called receptor-mediated transcytosis, or RMT. Specific receptors on the blood side of the endothelial cell bind to the molecule insulin or transferrin, are examples. This binding triggers the cell to engulf the receptor molecule complex in a vesicle, ferry it across the cell, and release the molecule on the brain side. It's a very selective, active transport system.
So highly controlled uptake for specific large molecules it recognizes.
Exactly. There's also something called absorptive-mediated transcytosis, AMT, which seems to work for some positively charged molecules. They might stick to the negatively charged cell surface and trigger uptake that way.
Okay, so basically, unless a drug is small and fatty, or it can somehow hijack one of these very specific natural transport systems, it's not getting into the brain easily.
That's the crux of it. And many promising new therapies, like gene therapies or protein drugs, are just too big or don't fit those transport mechanisms.
And that's where the magnetic nanoparticles come in, as a potential way to carry these therapies across.
Precisely. MNPs offer potential strategies to either force entry or sneak these otherwise excluded therapies past the BBB fortress.
Okay, so the BBB is this major obstacle. How exactly are researchers proposing to use magnetic nanoparticles and magnetic fields to get past it? What are the strategies?
The sources lay out several really clever approaches, leveraging both the particles and the fields. Strategy 1 is basically using a direct magnetic driving force.
Right, this is the idea of using the external magnet to literally pull the nanoparticle with its drug cargo across the barrier.
That's the concept. You apply a static magnetic field outside the body, focused on the brain region you want to target. The field gradient, how steeply the field strength changes over distance, creates a force on the magnetized nanoparticles in the blood vessels. The aim is to make this magnetic force strong enough to physically pull the particles through or between the endothelial cells of the BBB.
What determines how strong that pulling source is?
It mainly depends on two things: the magnetic moment of the nanoparticle itself, how strongly magnetic it becomes, and that magnetic field gradient. You want both to be high. But this force has to fight against the drag from the blood flowing past the particle.
Okay, so you need a strong magnet and strongly magnetic particles. Seems straightforward, but I sense a but coming.
Yes, there's always a trade-off, isn't there?
Yeah.
The sources highlight a key one here. You might think bigger particles would be better because they generally have a stronger magnetic moment, right? They feel the force more.
Makes sense.
But smaller particles, especially those ultra-small ones, the US pions under 50 nanometer, are much better at actually navigating through the tiny capillaries and potentially squeezing through biological barriers. Plus, their superparamagnetism is better for preventing aggregation when the field is off.
So small is good for getting through, but maybe not strong enough to be pulled effectively?
Exactly. The magnetic force scales roughly with the particle's volume, while the drag force scales more with its radius. So finding that sweet spot small enough to navigate and be stable, but big enough to respond well to the magnetic pull is a real challenge. There's likely an optimal size range. What are the other big hurdles with this direct pulling approach? A major one is physics. Magnetic field strength drops off very quickly as you move away from the source magnet. This makes it incredibly difficult to generate a strong enough field and a steep enough gradient deep inside the human brain. Targeting areas more than, say, 2 centimeters below the skull surface with this method alone is really, really tough.
Okay, so depth is a big problem.
Huge problem. Also, precisely focusing the magnetic force to accumulate particles only at your target spot is hard. Basic physics principles, like Earnshaw's theorem for static fields, make it difficult to create a stable magnetic trap. You might need complex, dynamic magnetic field systems to achieve even relative focusing.
And what happens when you turn the magnet off?
That's another issue. If the particles haven't been quickly taken up by the brain cells or lodged in the tissue, the blood flow can just wash them away the instant the magnetic pull stops.
Yeah.
So improving retention, maybe by designing particles that get internalized faster, is crucial.
So summing up this first strategy, direct magnetic pulling is probably more feasible for targets closer to the surface and maybe in areas where blood flow is slower, like capillaries.
That seems to be the consensus from the sources, yes. It's promising, but has significant limitations, especially for deep brain targets.
Okay, what's the second main strategy then?
This one is quite different. Instead of using the magnet to pull the particle across the barrier, it uses magnetic effects to temporarily make the barrier itself more permeable, to sort of open the door for a short time.
Ah, okay, so the magnet helps loosen the barrier and then the particles, or maybe other drugs, can get through more easily.
Exactly. And a key way to do this is through magnetic heating or mild hyperthermia using an alternating magnetic field, or AMF.
Alternating field. So not a static magnet pulling, but a field that's rapidly switching direction.
Right. When you expose magnetic nanoparticles, especially superparamagnetic ones, to an AMF, their magnetic moments try to flip back and forth rapidly to follow the changing field. This constant reorientation causes friction and energy loss, which generates heat locally, right around the nanoparticle.
How does that happen exactly? What's the mechanism?
For the espions we're mostly talking about, the main heating mechanism is usually something called Neal relaxation. That's the magnetic moment inside the particle flipping its orientation relative to the particle's crystal structure. There's also Brownian relaxation, which is the entire particle physically rotating back and forth in the surrounding liquid, creating viscous heat. But for many iomens in biological settings, Neal relaxation is thought to dominate.
Okay, so the AMF makes the particles heat up. How does that help with the BBB?
This is where it gets clever. You don't want to cook the brain, obviously. Very high heat, say above 42 degrees Celsius, can kill cells that's using cancer therapy. But if you use the AMF to generate just a mild controlled temperature increase, maybe to around 38 or 39 degrees Celsius, just a degree or two above normal body temperature, something interesting happens.
What's that?
This mild, localized hyperthermia seems to cause a reversible loosening of those tight junctions between the endothelial cells. It might also enhance the natural transcellular transport pathways. Basically, it temporarily makes the BBB slightly leakier, creating a window of opportunity.
Reversible being the key word there, hopefully.
Absolutely crucial. You want the barrier to recover fully once the AMF is off. But during that window, nanoparticles or maybe even other drugs circulating in the blood could potentially pass through the loosened junctions into the brain tissue more easily.
And I think you mentioned this heating could do double duty.
Yes. If the nanoparticle is designed with a temperature-sensitive coating, or if the drug itself is loaded in a way that it's released by heat, then the same AMF signal that helps open the BBB could also trigger the release of the drug payload once the particle has reached the target area.
That's very neat. Open the door and drop off the package with one signal.
Potentially, yes. The sources show research exploring both effects. Besides heat, they also briefly mention using magnetic forces, perhaps in combination with other things like focused ultrasound, FUS, to induce mechanical stimulation. The idea is that physical stress on the endothelial cells might create temporary pores, sometimes called magnetoporation, also aiding passage.
Are there other ways around the BBB that might work alongside MNPs?
Yeah, the sources touch on a couple. One is trying to bypass the bloodstream route altogether, like intranasal delivery. The idea is particles might travel along nerve pathways from the nasal cavity directly into the brain. Another point is that in some diseases, like brain tumors, the blood vessels within the tumor are often abnormal and already leakier than the normal BBB. So MNPs might have an easier time accumulating specifically in those leaky tumor vessels, which helps with targeting the tumor itself.
So the strategy really depends on where in the brain you're trying to go and what the condition is.
Absolutely. It's about choosing the right tool or combination of tools for the job.
Okay, let's shift gears a bit. We've talked a lot about the brain, but what about the rest of the nervous system? Damage to peripheral nerves, the ones in our limbs, face, etc., is a huge problem, causing pain, numbness, weakness. It's notoriously difficult to get these nerves to heal properly. Can magnetic nanoparticles play a role in peripheral nerve regeneration?
Yes, definitely. This is another area where MNPs are showing real promise, according to the sources. Repairing a severed or crushed peripheral nerve is complex. You need to encourage the surviving nerve cells to regrow their axons, guide those axons across the injury gap, and help them reconnect correctly with their target tissues, like muscles.
And how can MNPs help with that process?
In several ways, actually. Sometimes they act mainly as advanced delivery vehicles, but other times the magnetism itself is used actively.
Okay, let's start with delivery. What are they delivering?
Key therapeutic molecules that promote nerve growth and survival. A big one is nerve growth factor, or NGF. It's crucial for keeping neurons alive and stimulating them to extend their axons, or neurites.
How are MNPs used to deliver NGF?
Research examples include loading NGF into magnetic liposomes, fatty bubbles containing MNPs, or directly attaching NGF to the surface of gold-coated MNPs. Studies showed these approaches could enhance neurite outgrowth, sometimes even guiding the direction of growth when combined with a magnetic field.
What about delivering genetic material?
Yes, that too. Functionalizing MNPs to carry Surna, which can silence genes that might be inhibiting regeneration, or using polymer coatings like polyethylenamine to help MNPs carry plasmid DNA into cells, delivering genes that actively promote repair. They also mentioned using chetus-coated MNPs for delivery. There's even a suggestion that the slow release of iron ions from the MNP core itself inside the cell might have a direct stimulating effect on neurite growth.
So, providing the biological signals needed for repair, what about physically guiding the process? Does the magnetism come into play there?
It certainly does. This is the cell guidance aspect. If the cells involved in nerve repair, like the neurons themselves, or crucial support cells called Schwann cells, take up MNPs, then you can use an external magnetic field to actually apply a physical force to those cells.
You mean you can pull the cells where you want them to go?
Essentially, yes. By carefully shaping the magnetic field, researchers have shown they can guide the migration of Schwann cells towards an injury site, helping them form a bridge for axons to grow across. They've also guided the extension and controlled the orientation of growing neurites from nerve cells in culture. It's like using magnetic tweezers on a cellular level to orchestrate the repair process.
That's pretty incredible, using magnets to literally steer nerve regrowth.
It really is. And this potential is often combined with another approach, integrating MNPs into biomaterials. Researchers are creating magnetic scaffolds.
What are those?
They take materials commonly used to help bridge nerve gaps, things like nanofibers or hydrogels, and they embed MNPs within them. So you get magnetic nanofibers or magnetic hydrogels.
What's the advantage of making the scaffold magnetic?
Well, scaffolds provide physical support and structure for the regenerating nerve. Nanofibers can mimic the natural environment around nerves, while hydrogels are soft and can hold growth factors. Making them magnetic adds new capabilities,
How are the MNPs put into the scaffolds?
either by mixing pre-made MNPs into the scaffold material during fabrication, or sometimes by actually synthesizing the MNPs within the scaffold material itself. They do have to be careful to stop the MNPs clumping up inside the scaffold, often using those coatings or grafting techniques we talked about earlier.
What kinds of scaffold materials are used?
The sources mention things like chitosan, PVA, PCL, PLLA, HA, various biodegradable polymers often used in tissue engineering, as well as hydrogel materials like alginate and hyaluronic acid.
And how do these magnetic scaffolds help nerve repair?
They've been shown to significantly boost neurite outgrowth. Studies report, espions in hydrogels increasing axon elongation or espion grafted fibers, leading to maybe 20–30% longer neurites compared to non-magnetic controls. The magnetism seems to provide a beneficial cue.
Does the magnetic field play a role after the scaffold is in place?
Yes. You can use an external magnetic field to help align the structures within the scaffold as it's being made, or even after implantation. For example, creating aligned columns in a hydrogel can provide directional guidance for the growing nerve fibers, like tracks for them to follow. There was mention of a concept called Martini, using aligned magnetic fibers in a hydrogel specifically as a nerve interface.
So, aligning the physical structure,
Yep.
anything else?
Yes, just like with the DBB, you can use an alternating magnetic field, AMF, to gently heat the MNPs within the scaffold. This magnetocaloric effect can be used to trigger or accelerate the release of drugs or growth factors that were loaded into the scaffold, providing on-demand delivery right at the injury site.
That's incredibly versatile support, guidance, and controlled release. The sources also briefly touched on chemotherapy-induced peripheral neuropathy, CIPN.
Right, that's the nerve damage some people get as a side effect of cancer treatment. Interestingly, the sources noted some clinical studies suggesting that applying magnetic fields alone, not necessarily with nanoparticles, showed some promise in improving nerve function and reducing symptoms in CIPN patients.
So magnetism itself might have some direct therapeutic effect on nerves.
It's possible, though the mechanisms aren't fully understood yet. It suggests magnetism's role in neurology might be even broader than just nanoparticle delivery.
Okay, we've covered a lot of exciting potential, breaching the BBB, regenerating nerves, guiding cells. But whenever you talk about putting engineered nanoparticles into the body and applying external fields, the safety question looms large. Is this stuff safe? What are the toxicity concerns?
That's absolutely crucial, and the sources are clear that safety needs ongoing, careful investigation. While iron oxide nanoparticles are generally considered more biocompatible than many other types, the results aren't always consistent, and there's a definite need for more long-term studies, especially for chronic effects or use in sensitive areas like the brain.
How does the body typically handle nanoparticles anyway? How does it get rid of them?
Well, generally, they might be broken down through metabolic pathways if possible, or they get cleared out by the immune system, particularly cells of the reticular endothelial system like macrophages, which engulf foreign particles. But the concern, especially for the brain, is that accumulation or certain interactions could cause problems before they're cleared.
What are the specific risks associated with the iron oxide nanoparticles, the IONs?
The main worry is iron overload, or iron accumulation, particularly in the brain, which is very sensitive to having just the right amount of iron. Too much iron can be toxic.
How is it toxic?
Excess iron can trigger oxidative stress. It participates in a chemical reaction called the Fenton reaction, which generates nasty molecules called reactive oxygen species, or ROS-free radicals. These ROS can damage cells, DNA, proteins, lipids, basically cause havoc at the molecular level.
And oxidative stress is linked to brain diseases, right?
Yes, exactly. The sources specifically mention that iron-induced oxidative stress can promote the aggregation of proteins like amyloid beta, which is linked to Alzheimer's, and alpha-synuclein, linked to Parkinson's. So there's a theoretical risk that if IONs lead to significant iron buildup, they could potentially contribute to neurodegeneration. It's a major concern that needs careful management through particle design and dosing.
Could the iron also damage the BBB itself?
There's evidence suggesting high iron levels might do that too, yes. It could potentially inhibit key proteins, like occludin, that are essential for maintaining those tight junctions. So, ironically, the tool used to cross the BBB might weaken it if not used properly. It highlights the very delicate balance required.
Are some brain cells more vulnerable than others?
Possibly. The sources mentioned that glial cells, the brain's support cells like astrocytes, might take up ions more readily than neurons, potentially making them more susceptible to any toxic effects.
And what about how long the particles stick around? Is longer always better?
That's another trade-off. For drug delivery, you often want the particle to persist at the target site for a while to ensure the drug is released effectively. But that same persistence could increase the risk of toxicity if the particles aren't eventually cleared or broken down safely.
Does the coating or surface charge affect safety?
Oh, absolutely. The surface properties are critical. The coating can shield the core and reduce interactions, but the coating material itself needs to be safe. And surface charge matters too. Some studies suggest that high concentrations of nanoparticles with a strong positive or negative charge might negatively impact the cells of the neurovascular unit and potentially compromise the BBB. So careful surface engineering is key for both function and safety.
What about the external magnetic fields themselves? Are they safe?
There are established safety guidelines from bodies like ICNIRP and IEE for human exposure to magnetic fields, mainly concerned with preventing nerve stimulation or excessive tissue heating. The sources suggest that, based on modeling, the field strengths needed for MNP applications likely fall within a safe margin below these external limits. However, the potential long-term biological effects of applying these specific types of localized, potentially strong or rapidly changing fields for therapeutic purposes still need thorough investigation.
So the bottom line on safety is iron oxides are relatively good candidates, but iron overload and oxidative stress are real concerns, especially for the brain. Everything from the particle's core and coating to the dose and the magnetic field needs careful optimization and long-term study.
Precisely. Safety isn't an afterthought. It has to be designed in from the very beginning.
Okay, so given this incredible potential, but also these significant safety and technical considerations, what are the main hurdles still standing in the way of seeing these magnetic nanoparticle therapies widely used in clinics? What's the focus for future research?
The sources point to several key challenges that the field is actively working on. Technically, a really big one is achieving precise magnetic focusing deep within the human body.
We talked about the field dropping off with distance.
Exactly. Scaling up from, say, a mouse brain where the target is millimeters deep to a human brain where it might be many centimeters deep while maintaining sufficient force or heating effect and precise localization. That's a massive engineering challenge for the external magnet systems.
Right, the physics is tough. What else?
Improving particle retention at the target site. Remember the old washout problem. If the particles don't get taken up by cells quickly after the magnetic field guides them there, they might just leave with the blood flow. So, designing particles that are internalized more efficiently and specifically by the target cells is crucial.
Especially important if the effect relies on intracellular action.
Definitely. And specifically for the BBB applications, developing really reliable and safe methods to control the temporary opening. How do you ensure it opens just enough for just long enough and then closes completely without any lingering damage or side effects? That requires exquisite control and monitoring.
Monitoring. That implies imaging, right?
Yes. Another challenge is developing better ways to see exactly where the nanoparticles are in the body in real time during treatment. Good in vivo imaging is needed to confirm they've reached the target and are staying there at the right concentration.
Those sound like significant technical mountains to climb. Are there other kinds of challenges?
Yes, the sources also really emphasize the interdisciplinary nature of this work. This isn't just a physics problem or a chemistry problem or a biology problem. It's all of them woven together. Meaning, you need physicists who understand magnetism, chemists building the particles, biologists understanding the cellular responses, pharmacologists dealing with the drug aspects, engineers designing the devices, and clinicians identifying the real medical needs and translating the tech safely. Success requires all these experts working together seamlessly.
Like a task force approach, as one source put it.
Exactly. For any specific disease target, you need this collaborative team to figure out the entire system. The best MNP type, the right dose, the ideal functionalization, the drug cargo, the delivery route, the best way to cross barriers like the BBB, and how to minimize toxicity, all at the same time.
It's not just about having a cool nanoparticle, it's about the whole integrated therapy.
Precisely. The clinical need has to drive the design. And the sources highlight that animal studies and sophisticated computational modeling are going to be vital tools for designing and testing these complex systems before they can move towards human trials.
Okay, this has been a truly deep dive into a complex field. Let's try to pull back and maybe summarize the key takeaways about these magnetic nanoparticles in medicine.
Sounds good. So first off, we learned they are essentially tiny magnets with big potential. MNPs, especially those superparamagnetic iron oxides, SPONs, have unique magnetic abilities at the nanoscale, crucially avoiding clumping when no field is applied, which is vital for use inside the body.
Right. And they are meticulously engineered for purpose. Scientists synthesize them with incredible control over size and shape, then coat and functionalize them. This makes them stable, biocompatible, and capable of carrying specific drugs or targeting molecules.
Which enables targeted treatment. They can act as vectors, guided by external magnetic fields to deliver therapies precisely where needed. Or their magnetic properties can be used to trigger controlled drug release, for instance, through magnetic heating.
A huge area of potential is opening barriers, especially that tough blood-brain barrier. Magnetism offers ways to tackle this, either by physically pulling particles across or by temporarily increasing the barrier's permeability using heat or mechanical forces.
And we saw exciting applications in repairing nerves. MNPs are being explored to boost peripheral nerve regeneration by delivering growth factors or genes, by magnetically guiding repair cells, and by enhancing the properties of biomaterial scaffolds used to bridge nerve gaps.
But, importantly, safety is always in focus. While iron oxides are relatively biocompatible, potential issues like iron accumulation, oxidative stress, and effects on barrier integrity need careful consideration and long-term study. Particle design, dose, and clearance are critical safety parameters.
Agreed. And finally, the road ahead involves tackling significant technical hurdles, like deep magnetic focusing and particle retention, but also requires overcoming collaborative challenges by building strong interdisciplinary teams to design optimal, safe, and effective therapies for specific diseases.
It really underscores that this isn't just about one scientific discovery, but about integrating knowledge from many fields. So let's leave our listeners with a final thought to ponder. If we can truly master using invisible magnetic forces to guide these incredibly tiny particles deep within the body, targeting specific cells or tissues with precision, maybe even combining diagnosis and therapy, what other kinds of precise, non-invasive medical interventions might become possible down the line that we can barely even imagine today?
That's a fascinating question. The potential really does feel vast. If you can manipulate things reliably at that scale, guided from outside, it could open doors to entirely new ways of interacting with biology for therapeutic benefit, beyond just delivery, maybe influencing cell behavior directly.
The possibilities seem immense. The research we've discussed today really paints a picture of a future where magnetism and nanotechnology could revolutionize treatment for some of our most challenging medical conditions. Thank you for joining again for this deep dive.